Good morning, everyone. Today we will start with a new topic of the chapter Photosynthesis in Higher Plants of class 11th biology. The name of the topic is Mechanism of Photosynthesis. The main objectives that we will cover today in this video includes mechanism of photosynthesis. Here we will discuss the first phase of photosynthesis, that is light phase, which includes photolysis of water, photophosphorylation and chemiosmotic hypothesis. Mechanism of photosynthesis. As already discussed, photosynthesis is the process by which green plants prepare their own food in the presence of sunlight and chlorophyll by utilizing carbon dioxide and water. This process of photosynthesis occurs in the mesophyll cells of leaves. During daytime, the chlorophyll molecule traps solar energy. This trapped energy is utilized for the synthesis of organic compounds in the form of glucose using carbon dioxide and water as inorganic ingredients. Oxygen gas is evolved as a byproduct of this process. It involves two steps: light reaction or the photochemical phase, and the second is dark reaction or the biosynthetic phase. Light reaction. This process occurs in the thylakoids of chloroplast in the presence of sunlight, hence its name light reaction. It is mainly concerned with the formation of NADPH, a reducing power, and adenosine triphosphate. These compounds constitutes the assimilatory power. Another end product formed as byproduct is oxygen. Light reaction is also called as Hill's reaction, as the process of light reaction was explained by Robert Hill. It is also known as photochemical phase, as in light reaction, light energy is converted into chemical energy. Dark reaction or biosynthetic phase. In dark phase, carbon dioxide is reduced to glucose by means of energy through ATP and reducing power through NADPH that are produced during light reaction. For dark reaction, there is no need of light, hence it is called as dark reaction. This process occurs in the stroma of chloroplast. This process is also called as Blackman's reaction, as it was explained by the scientist Blackman. It is also known as biosynthetic phase because here the chemical energy produced during light reaction is utilized to convert or to fix carbon dioxide to form organic compound, that is glucose. Now let's discuss the steps of light reaction. Light reaction includes following steps. The first step is excitation of chlorophyll. The chlorophyll on exposure to light absorbs radiant energy as photon and becomes activated. Next is photolysis of water. Here photo word means light and lysis means splitting. So photolysis is actually the splitting of water molecule into hydrogen and hydroxyl ions with the help of light energy. The radiant energy from sunlight is absorbed by chlorophyll and it is transferred into chemical energy. This chemical energy is capable of splitting water into hydrogen and hydroxyl ions. The next step is production of NADPH or reducing power. Photolysis of water produces hydrogen and hydroxyl ions. Hydrogen ions are immediately picked up by NADP, activated by electrons. It results in the formation of reducing power NADPH, that is nicotinamide adenine dinucleotide phosphate. Hydroxyl ions produced during the process of photolysis of water undergoes oxidation to form highly unstable compound called hydrogen peroxide, which readily splits to form molecular oxygen and water. Hence, we can say that oxygen produced during the process of photosynthesis comes from water, not from carbon dioxide. The next step is photophosphorylation. Here, photo word stands for light, phospho word stands for phosphate, and rylation means process of formation. So, photophosphorylation is defined as the process of formation of energy-rich ATP from ADP and inorganic phosphate. The electrons available in the above reaction are picked up by certain electron carriers. When the electrons pass through these carriers, they dissipate energy. This energy is utilized in the formation of energy-rich compound ATP from ADP and inorganic phosphate. Now we'll discuss these steps in detail one by one. Light reaction. During the course of light reaction, light is trapped by photosynthetic pigments present in the quantosome of grana thylakoids. These photosynthetic pigments are organized into discrete photochemical light harvesting complexes known as photosystem 1 and photosystem 2. Both these photosystem 1 and photosystem 2 consist of two types of pigment: accessory pigments and essential pigments. Essential pigments are the pigments which are directly involved in the process of photosynthesis. They take their excitation energy from accessory pigments. Accessory pigments are the pigments which are not directly involved in the process of photosynthesis but are supporting in nature. They trap the sunlight directly and pass it to essential pigments. Essential pigment includes chlorophyll B, chlorophyll A, whereas accessory pigments includes carotenoids, xanthophylls, etc. Photosystems. Light harvesting complexes or photosystems are made up of hundreds of pigments molecules bounded by proteins. Each photosystem has a photocenter or reaction center which is actually representing essential pigment where actual reaction takes place. This reaction center contains a special chlorophyll molecule and is fed by 100 other pigment molecules forms the light harvesting system called antennae. Antennae molecule absorb light of different wavelength but shorter than that of reaction center in order to make photosynthesis more efficient. Then these antennae pass the light energy to reaction center. These photosystems are named according to the sequence of their discoveries, not in the sequence in which they function during light reaction. The reaction center is different in both the photosystems as given below. In photosystem 1, the reaction center or chlorophyll A has a peak of absorption at 700 nanometer and is known as P700. In photosystem 2, the reaction center has absorption peak at 680 nanometer. Hence, it is called as P680. If we talk about photosystem 1, it is involved in the cyclic and non-cyclic photophosphorylation. Photosystem 1 receives the electron from photosystem 2. This system produces a strong reductant which reduces NADP to NADPH2. Molecular oxygen is evolved in this system. Photosystem 1 is located in the outer surface of thylakoid, that is, appressed regions of grana. The reaction center of photosystem 1 is P700. Chlorophyll carotenoids pigments are in ratio of 20:30:1. Chlorophyll A with maximum absorption at 700 nanometer is acting as a reaction center. Now if we talk about photosystem 2 is involved only in non-cyclic photophosphorylation. Photosystem 2 donates electron to photosystem 1 where NADP is reduced. This system is responsible for the photolysis of water and involves the evolution of molecular oxygen. Photosystem 2 is located on the inner surface of the thylakoid, that is, in the stroma and non-appressed regions of grana. The reaction center of photosystem 2 is P680. Chlorophyll carotenoid pigments are in ratio of 3:7:1. Chlorophyll A with peak absorption at 680 nanometer is acting as its reaction center. Photolysis or splitting of water. As already discussed, photolysis is the splitting of water in the presence of light. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. The photosystem 2 in turn regains the lost electrons by the photolysis of water. You can see here water molecule is splitting into hydrogen and hydroxyl ions along with the release of 4 electrons. While all the electrons formed are replaced, the proton gets accumulated in the lumen of thylakoid and the oxygen gets into the atmosphere. Electron transport. The photosynthetic electron transport chain initiated by the absorption of light by the photosystem 2. The red light of wavelength 680 nanometer is absorbed by the reaction center of photosystem 2, due to which electrons becomes excited and jumps into an orbit away from the atomic nucleus. These electrons are then picked up by electron acceptors, which passes them further to electron transport system consisting of cytochromes. It is to be noted that this movement of electrons is downhill according to the redox potential scale, that is, oxidation reduction scale. The electrons of the electron transport chain are not used up in the chain, instead, they are further passed on to the pigments of photosystem 1. Now, like photosystem 2, electrons in the reaction center of photosystem 1 also gets excited on receiving red light of wavelength 700 nanometer and gets transferred to the another electron acceptor with high redox potential. The electron in this case also moves downhill, but this time the electrons does not move to a reaction center or chlorophyll A, instead moves to a molecule rich in energy and NADP. On addition of these electrons, the NADP gets reduced to NADPH and hydrogen ions. In 1960, Bandel and Hill discovered the Z scheme of electron transport. It is a series of reactions that we have just studied above from the whole scheme of electrons transport, initiating from photosystem 2 uphill to the acceptor molecule, down the electron transport chain of photosystem 1, excitation of electrons, and then their transfer to another acceptor, and finally downhill to NADP in order to get reduced to NADPH and hydrogen ion. Photophosphorylation is the process through which ATP is synthesized from ADP and inorganic phosphate by the cell organelle like mitochondria and chloroplasts with the help of energy from solar radiation. It refers to the process in which ATPs gets formed from ADP in the light reaction of the process of photosynthesis. Photophosphorylation in mitochondria is not light dependent, but it uses the energy by oxidation of nutrients to produce ATP. Hence, it is also called as oxidative photophosphorylation. Arnon and his co-workers were first to show that isolated chloroplasts can produce ATP when exposed to light. That is the process of photophosphorylation. This ATP production occurs through electron transport chain, which is of two types: non-cyclic photophosphorylation and cyclic photophosphorylation. Non-cyclic photophosphorylation. It is a type of photophosphorylation in which both the photosystems, photosystem 1 and photosystem 2, cooperate in light-driven synthesis of ATP. During this cycle, electrons released from photosystem 2 does not return to it. Hence, it is known as non-cyclic photophosphorylation. Both NADP and ATP are formed during this reaction. The light energy of specific wavelengths below 680 nanometer are absorbed by pigment molecules of photosystem 2. They become excited and transfer their energy to reaction center P680 of photosystem 2. As a result, P680 releases electrons accepted by primary acceptor, probably pheophytin. P680 is then oxidized and is again reduced by receiving electrons from the oxidation of water. You can see here water is oxidized in the presence of light energy. This reaction is called as Hill's reaction, which showed that oxygen comes from water with the formation of hydrogen ions and electrons. Hydrogen ions are required for the reduction of NADP to NADPH, and energy of the electron is used for the formation of both NADPH and ATP. This photolysis of water requires manganese and chlorine ions as a cofactors, which is indicative of some enzyme involved in it. This oxidation of water takes place in 4 steps and each requires one enzyme. The reduced pheophytin then passes its electrons to various electron carriers of electron transport chain in sequence. The electron goes from pheophytin to plastoquinone, which then passes these electrons to cytochrome complex, which includes cytochrome B6 and cytochrome F, and then to plastocyanin, which ultimately hand over these electrons to photosystem 1. The formation of ATP takes place when the electron passes from plastoquinone to cytochrome F. 12 molecules of ATP are formed in non-cyclic electron transport. These electrons then leave P680 and then do not return back to it. Hence, it is called as non-cyclic photophosphorylation. From P700, the electrons are accepted by A1, then A2, and then A3. These A1, A2, A3, these are unknown electron acceptors. And then ultimately electron reaches to ferredoxin. Now, the reduced ferredoxin transfers its electron to NADP, which gets reduced to NADPH and hydrogen ion in the presence of NADP reductase. Here we can see that photosystem 2 is associated with photooxidation of water, the production of 12 molecules of ATP through non-cyclic transport, reduction of NADP to NADPH and hydrogen ions released in photooxidation of water. In the formation of NADPH, photosystem 1 is also involved. It can be understood that formation of NADPH is a function of photosystem 2, but it is generated through photosystem 1. Cyclic photophosphorylation. It is a type of photophosphorylation in which only photosystem 1 is taking part and the electrons released from the reaction center, that is P700, returns back to it after passing through a series of carriers, that is circulation takes place within the photosystem and phosphorylation occurs due to cyclic flow of electrons. This transport is active in plants when NADPH starts accumulating and carbon dioxide assimilation is checked. It helps in the production of 6 more ATP molecules. In this, electrons released by P700 finally come back to photosystem 1. Hence, this cycle is called as cyclic electron transport. It does not require an external source of electrons. There is no photolysis of water. It occurs mostly in stromal or intergranule thylakoids. It is not inhibited by dichlorophenyl dimethylurea. It operates under high light intensity, anaerobic conditions or under poor carbon dioxide supply. The pigments of photosystem 1 absorb specific wavelengths of light beyond 680 nanometer and becomes photoexcited. After photoexcitation, electrons are released and transferred to the reaction center of photosystem 1. The electrons from P700 reaches to ferredoxin via A1, A2 and A3 as in case of non-cyclic transport. From reduced ferredoxin, these electrons reaches to plastocyanin via cytochrome B6. After this, electron finally reaches to P700. After plastoquinone, the path of electron is similar to non-cyclic transport. In this cyclic transport, the ATP formation takes place when electrons are passed from reduced plastoquinone to cytochrome F. As per earlier report, ATP formation could also take place between reduced ferredoxin and cytochrome B6. It becomes evident that in green plants, the non-cyclic electron transport chain is dominant. The two photosystems are connected to each other through an electron transport chain giving an appearance of Z. Robert Hill and Bendel called it as Z scheme. When non-cyclic form of photophosphorylation gets stopped, under certain conditions, the cyclic photophosphorylation occurs. The cyclic photophosphorylation takes place in the stromal lamellae of chloroplast. This happens because the stromal lamellae does not possess enzyme NADP reductase, essential for reducing NADP to NADPH and photosystem 2. Thus, the excited electrons in the cyclic photophosphorylation does not pass on to NADP. Instead, it gets cycled back to photosystem 1 complex. chemiosmotic hypothesis and synthesis of ATP molecule. This hypothesis was given by Mitchell in 1961. It explains the mechanism of ATP synthesis within the chloroplast during photosynthesis. During the photochemical phase or light reaction, ATP and NADPH are generated. These are the key components and used in dark reaction for the production of the final product of photosynthesis. Let us see how this ATP and NADPH are generated during the light reaction. ATP synthesis is linked to development of proton gradient across the membrane of thylakoid and mitochondria. The process that causes development of proton gradient across the membrane is photolysis of water, that is splitting of water molecule that occurs inside the thylakoid to produce hydrogen ions or protons. As electron passes through photosystems, protons are transported across the membrane because primary acceptor of electron is located towards the outer side of membrane. The NADP reductase enzyme is located in the stroma side of membrane. Photolysis of water molecule takes place on the inner side of thylakoid membrane in photosystem 2. The hydrogen ions or protons that are produced by the splitting of water accumulates within the lumen of thylakoid. As the electron moves through the photosystem, protons are transported across the membrane. It is due to the presence of primary acceptor of electrons located towards the outer side of the membrane and transferring its electron not to electron carrier but to an hydrogen carrier. Therefore, this molecule removes a proton from the stroma while transporting an electron. When this molecule passes an electron to the membrane, the proton is also released into the inner side of the lumen. The NADP reductase enzyme is located on the stroma inside the membrane. For the reduction of NADP to NADPH, electrons comes through photosystem 1 and protons required comes from the stroma. Due to this, within the chloroplast, proton in the stroma decreases in number and in the lumen there is accumulation. It creates a proton gradient across the thylakoid membrane. By the breakdown of this gradient, energy is liberated. Breakdown refers to the movement of proton by the coupling factors of ATPase across the membrane to the stroma. This energy activates ATPase enzyme and catalyzes the formation of ATP from ADP and inorganic phosphate. So in brief, we can say that in plants during light reaction, photosystems help chlorophyll to absorb light. As a result, hydrolysis takes place and releases electrons and protons. The electrons get excited to higher energy level and are transported by the electron transport system, while protons from the stroma starts to accumulate inside the membrane. This creates a proton gradient. Some protons are used by photosystem 1 for reduction of NADPH. When the proton gradient is collapsed, it releases energy and protons are carried out back to the stroma via F0, the transmembrane channel of ATP synthetase. This release energy causes changes in F1 configuration and triggers the ATP synthetase to convert ADP to ATP. The proton gradient is important as it is the breakdown of this gradient that leads to release of energy. This gradient gets broken due to the movement of protons across the membrane through transmembrane channels of F0 of the ATPase towards the stroma. In this video, we have completed the first phase of photosynthesis, that is light phase. In next video, we will start with the next phase of photosynthesis, that is dark phase. Thank you everyone.